So thank you, Martha. So we'll keep questions for later. I will move on to the second talk, which will be presented by Jake Meyers from Texas A&M University.
Okay. All right, so I'm going to be talking about our current progress on a project looking into the genetic basis for outdoor host seeking in Anopheles coluzi mosquitoes from Bioko Island, Equatorial Guinea. So Bioko Island is located off the western coast of Central Africa. Being located so near to the equator, it experiences year-round hyperendemic malaria transmission. Historically, this has resulted in very high Plasmodium prevalence among children, as well as a very high infant mortality rate. In 2004, the Bioko Island Malaria Control Project was founded as a partnership between the government of Equatorial Guinea, Medical Care Development International, and the Marathon Oil Corporation. Their goal was to reduce and eventually eliminate malaria from Bioko Island. And a major arm of this project was a large vector control program where they conducted annual or biannual indoor residual spraying of bendiocarb or deltamethrin from 2004 to 2015. And more recently, they've changed their approach to a stratified targeting approach where they would conduct IRS in regions with high Plasmodium prevalence, as well as doing an island-wide bed net distribution with nets impregnated with pyrethroids and the synergist PBO. So after 2 years of vector control, this vector control program had successfully eliminated 2 of the 4 vectors present on Bioko. And then in 2005, an operational grant from MCDI was given to us to measure the impact of vector control on the mosquito populations of Bioko Island. And we used population genetic approaches to estimate the effective population size before and after vector control. So in the first project, it was a longitudinal project to compare the estimated population size in 2004, before the Bioko Island Malaria Control Project, to 2009, and we discovered a 79% reduction in the estimated population size. A second project looked more closely at the effect of individual IRS spray rounds on these mosquito populations, and the second project was done on Anopheles coluzi, present in Mongola, which is in a village on the northern end of Bioko Island. And we saw these dotted lines denote the IRS spray rounds. And we can see that with each spray round, there's a significant decrease in the mosquito population, which eventually rebounds and then is knocked down again with subsequent IRS sprays. So it's clear from this data that the IRSs have a major impact on the Anopheles coluzi populations on Bioko Island, and that these spray rounds exert a large selection pressure on these mosquitoes. So in 2009, the vector monitoring program with the BIMCP was greatly expanded. From 2004 to 2009, they used exit window traps to capture mosquitoes that had fed indoors and were trying to escape early in the morning. But they were finding that these traps really weren't catching a lot of mosquitoes anymore. So they changed to a paired indoor-outdoor human landing catch scheme. This was conducted in these 4 villages. There were 8 collectors per village. These collections were done twice monthly, every month of the year. So initially, we were not expecting to find many mosquitoes host seeking outdoors. Anopheles coluzi is known to be very endophilic and prefer to feed on people indoors. So in the first year, we were very surprised to see a large proportion, and we're looking at proportions here, of indoor and outdoor host seeking mosquitoes. We see a large proportion preferring to host seek outdoors. We monitored this behavior through these paired indoor-outdoor HLCs over the years, and we saw that this preference to feed outdoors increased. So we analyzed this data using generalized linear mixed models, and we used those models to account for confounding variables such as collector bias, changing in housing condition over time, and environmental and seasonal factors that would happen over the years. So with this GLMM, we discovered an odds ratio of about 1.2, meaning that an individual mosquito is 1.2 times more likely to host seek outdoors versus indoors over time. So with this data, we were asking now, could this shift in host-seeking behavior represent a behavioral adaptation to these indoor applied insecticides? Or said differently, does outdoor host-seeking behavior have a genetic basis? So to answer this question, we're using a pool-seq design to do a genome-wide association study. So of the wild Anopheles gambiae population on Bioko, we sample the population using our paired indoor-outdoor HLC. Those are sent to us, and then we do DNA extraction of each individual and create pools with equimolar contributions from each individual, which we then put through library preparation and next-generation sequencing. We then use the reads from the next-gen sequencing and align them to the genome and count the alleles at each locus to get an estimate of the allele frequency. And we use that estimate as an estimate of the allele frequency for the actual population, from which we can calculate FST, which is a fixation index that varies from 0 to 1, and we can calculate that at each SNP across the genome. So the groups we're looking at, if we start with these post-intervention ones, we have the indoor and outdoor collected mosquitoes, which began in 2009, and we have another sample that we're looking at from 2014. And when we compare these samples, we're looking for polymorphisms that might distinguish these two phenotypes. Additionally, we have samples over time in which we can look for markers of selection, which we might not be able to see within a single year if some of these markers have gone to fixation. So this outdoor feeding phenotype we think is kind of a tricky thing. We've been studying these mosquitoes on Bioko for a long time, and we know they're not separate populations. And we think of this phenotype more as a tendency or preference to feed indoor versus outdoor. So just because an individual mosquito might have a preference to feed outdoors, we don't think that they would feed exclusively outdoors. And I think, or we believe that this would make our data fairly noisy and make the differences between our pools and between our phenotypes very small. So to detect these very small potential differences in allele frequencies, we're kind of throwing everything at this. So we have very large pool sizes, and we're sequencing our data to very high coverage. So the first analysis we're looking at right now, we're going to look at is the 2009 indoor versus outdoor. And we just got this data off the servers last week, so it's still somewhat preliminary. It's very preliminary, but we can take a look. So our analysis came up with over 15 million SNPs, which corresponds to about 1 SNP every 30 base pairs. So we can just see how they vary across the genome. And really the first issue or the first question is, how do we determine which of these SNPs are significant? And we've developed 2 methods. The first is a simulation-based method. So here, we're using simulations to predict the distribution of allele frequencies caused by pooling and sequencing individuals collected from the same population. And then we can take those allele frequencies, those ranges of allele frequencies, and calculate the FST to get a threshold for what we could expect to see from a single population. So to kind of work through an example, if we have a population with a starting allele frequency of 0.5, and we take a sample size from that population of 1,009 individuals, which is what we took for 2009, we would expect to see, and we simulate that sampling 10 million times, we would expect to see this distribution around 0.5. Now, if we take that distribution of allele frequencies and sequence it to a coverage of 500x, we would then expect to get allele frequencies, a distribution as such, around 0.5. Now, if we were to take 2 individual allele frequencies from this distribution at random and calculate FST and conduct that 10 million times, we get this distribution of FST values. And the maximum here we see is 0.06. So we decided to use this as a threshold given these parameters of coverage and allele frequency when we analyze our individual SNPs. So if we look at how the simulation works with coverage, we see that our ability to detect low FSTs really decays as coverage decreases, as we might expect. Additionally, with lower starting allele frequencies, we have more power to detect lower FST values. So we can fit these data with an exponential curve, and it fits very well. But what we want to be able to do is we want to take our SNPs, using their allele frequency and coverage, be able to determine across all of these coverages and allele frequencies whether or not they are significant. So we did a multivariate analysis containing both allele frequency and coverage and fit it to a 3D plane. So we can see again, as coverage decays, we get an increase in our ability, or an increase in the threshold of our ability to detect a significant SNP. And as allele frequency increases, we also get an increase in that ability, or kind of, I guess, a decrease in power to detect SNPs. So when we fit our data to that plane, out of our over 15 million SNPs, we had 10 hits that were positive, or that were significant. And so we haven't had time, like I said, we just got this data about a week ago. We haven't had time to go through all of these SNPs, but what really stood out was this small cluster on 3L that were all very near each other. So we've explored this. So it was found on this location, or in this region of 3L, and we can see several genes within this region with PGI found in the middle. And when we map our FST across this gene, we see a peak here in exon 2 within the coding region. PGI is a metabolic enzyme involved in glycolysis, and we'll kind of come back to that later. So we've looked at some of the top SNPs within this peak for synonymous versus non-synonymous mutations, and we found one non-synonymous SNP, which caused a mutation from serine to threonine. So this gives us some evidence, and this is our top candidate right now for maybe a distinguishing gene between indoor and outdoor host-seeking phenotypes. So our second method was a comparison of replicates within phenotype. So in our 2009 samples, we created two biological replicates from each phenotype, which had their own independent library prep and then were sequenced. We then took the top 100,000 SNPs from the comparison of each replicate, which constitutes under the top 1% of SNPs. And then we looked to see which of these SNPs overlapped between the two replicates. We found about 1,800 SNPs. And again, I haven't had time to go through all of these SNPs, but what we did find is we found a large cluster again in the PGI gene that we just looked at before. And we found another large cluster here on 3R. So we decided to explore this a little further. And so we're going to look into this region of 3R, and there are several genes here with GAPDH in the middle. And again, when we look at the FST values of our individual reps, we see a peak here in the coding region of the only exon that makes up this gene. GAPDH, which is glyceraldehyde 3-phosphate dehydrogenase, is another metabolic enzyme involved in glycolysis. Once again, we looked at the top SNPs within this peak for synonymous versus non-synonymous mutations. We found a non-synonymous SNP which caused an asparagine to lysine mutation. So just a bit more about these genes. Both PGI and GAPDH are part of the glycolysis pathway. And insect flight requires really very high rates of glycolysis and carbohydrate metabolism in the muscle tissue. Specifically, initiation of flight requires activation of glycolysis pathways in flight muscles, and this has been seen across a diverse range of arthropods. Now, PGI has been even been thoroughly studied as an allozyme and a neutral marker in various arthropods. And more recently, it was found that selection actually occurs on this gene due to, and this was seen in several butterflies, that selection occurred on this gene due to fluctuating daily temperatures. They found that heterozygotes had advantages over homozygotes across suboptimal temperatures, and that there was a directional selection along thermoclines. So this is all very preliminary, and it's still very unclear to us how PGI and GAPDH may directly influence indoor versus outdoor host-seeking phenotypes. But we believe, like with this background, we have some interesting associations and interesting behaviors associated with these genes, and they're candidates that we want to explore further. So we've just started as well looking at comparing the 2004 versus 2009 pools, and this is data we just got off the servers on Monday, so it's really brand new. The first thing I looked at were PGI and GAPDH, as you might imagine, and we were pleased to see that we see peaks in FST in both of these genes, in the same regions of these genes as well, so giving us more evidence that these candidates may be involved in this phenotype. So beyond that, I kind of wanted to do a quick survey across the chromosomes just to see if there was anything else really interesting. So I quickly just filtered out all of the low coverage SNPs and then looked for clusters within the top 25 SNPs on each chromosomal arm. And I wasn't really expecting to find much, But kind of surprisingly, we found clusters in 3 genes. So the first 2 genes are beta-tubulin and beta-actin. These are cytoskeletal elements. They form microtubules and microfilaments and are highly expressed in muscle. These microtubules and microfilaments make up the actual muscle fibers that are responsible for muscle contraction. So it's more evidence pointing towards the motor systems or something happening in the muscle tissue. The third gene was this Rho-type GTPase. Again, we see a peak. This is the first example where we see a peak in an intron. This is an exon here, a very small exon within the gene. But this is the first example where we see a peak within an intron of our candidates. So obviously we don't expect this to create any sort of mutation, but maybe it has some effect on expression. Rho-type GTPases, I haven't done very much literature review on this. There wasn't much in insects, but it was shown in mammalian smooth muscle cells to promote actin and myosin contraction. So more evidence pointing towards muscle, maybe metabolism, having some sort of influence on our phenotype. So the take-home points are that our results are very preliminary, but we are finding clusters of significant SNPs in the coding regions of PGI and GAPDH between indoor and outdoor host-seeking phenotypes. And the function of these genes really point towards a possible role for metabolism and maybe environmental temperature to be driving changes in their flight activity. So future directions. So we want to look for regions of the genome under selection, and we'll be using Tajima's D and Pi to look at this, and that's currently running on our servers. We need to finish our analysis of and get our 2014 samples sequenced, and we're hoping that this really validates these candidate genes we're seeing in 2009. And then we would like to genotype our candidate genes from individuals of each phenotype to look at their haplotypes, as well as perform potentially functional analysis of candidate genes. So that, I'd like to thank the members of the Slotman Lab, as well as collaborators at Texas A&M, Medical Care Development International, and London School. The funding for the vector monitoring and the mosquito collections was done through MCDI and the Bioko Island Malaria Control Project, and the funding for the genomic analysis is through NIH. Thanks for your time and attention.